Hello and welcome to biology class. I am Shipra from ADARSH MANDIR SHANKAR for class 11. Today we will start a new chapter, that is photosynthesis in higher plants. So first you have to know what is photosynthesis. Photosynthesis is the process of making food in the plants with the help of carbon dioxide, water in the presence of sunlight and chlorophyll, and the product of photosynthesis is carbohydrate, glucose, and oxygen is liberated in the form of byproduct. So it is the photosynthesis process in which carbon dioxide is available in the air, water is absorbed by the roots from the soil, sunlight is the ultimate source of energy, chlorophyll pigments are the green pigments which are present in the green parts of the plant like leaves. Okay, so this is the photosynthesis, but photosynthesis is not a short reaction, it's a very vast reaction. So first we are studying about the contribution of some scientist, Joseph Priestley. Joseph Priestley in 1770 he did a series of experiments that revealed the role of air in the growth of plant. So this is the very famous experiment, bell jar experiment. Do you know what is bell jar? Now see, this is the bell jar. It's a glass tumbler. So what he did, he just keep a candle with mouse, burning candle with mouse, and put it on the flat surface. So after some time, the candle and mouse, you can see here, this is the bell jar, this is bell jar, and this is candle, and this is mouse. So what happens? First he keep a burning candle with mouse in the bell jar. After some time, what he saw, that the candle is also now not burning and mouse is also died. Why? Because the oxygen which is present inside the bell jar, it is finished. So the both are, the burning process is also stopped, the burning of candle is also stopped, and the mouse is dead. But what he did next, he did keep a plant, a fresh plant, mint plant in the bell jar with the burning candle and mouse. Okay, now after some time, what happens? There will be no stopping the burning, candle is continuously burning, and mouse is also living. Why this happens? Because during the process of photosynthesis, the plant, it consumes the carbon dioxide and release oxygen during the photosynthesis. But these mouse, during the respiratory process, during the respiration, it consumes the oxygen and release carbon dioxide. Okay, so the carbon dioxide and oxygen balance is maintained through the mint plant and mouse. Okay, so this is the experiment of Priestley. And now the next is Gale in Gelhaus. Gale in Gelhaus showed that the sunlight is essential for the photosynthesis. And it is also shown that the oxygen gas is evolved during the photosynthesis process. Now, where the photosynthesis takes place? The process of photosynthesis takes place in the chloroplast. This is the structure of chloroplast. It is present in the green part of the plant like leaves. So what is the structure? This is surrounded by two unit membrane, that is outer and inner unit membrane, which is composed of lipid and proteins, and the liquid present inside the chloroplast is stroma. In the stroma, the dark reaction of photosynthesis takes place, and these are the thylakoids, the sac-like structure. These are the thylakoids, and the bundle of thylakoids are known as the granum. Granum is the site of light reaction, or we can say the Hill reaction. Hill is the name of scientist who gave it. Now these are the thylakoids, and on the thylakoid surface the chlorophyll pigments are present. The green pigment, chlorophyll pigments are present. Approximately 230 chlorophyll pigment per thylakoids present on the thylakoids, and 70 type of ribosomes are also present, and a circular DNA is present in the chloroplast. So this is the structure of chloroplast and the site of photosynthesis. Actually, photosynthesis process is divided into two phases, that is light reaction and dark reaction. Light reaction is also known as the Hill reaction. This takes place on the granum, and dark reaction takes place in stroma. Okay. Now we are starting the light reaction. So light reaction we can divide into two phases, that is cyclic photophosphorylation and non-cyclic photophosphorylation. The non-cyclic photophosphorylation Z scheme, it is also known as Z scheme, and during the Z scheme, what happens? The two reaction centers are there. These are the reaction centers, and reaction center it may be P700 and P680. So what happens? The photons, in the form of photons, the sunlight comes by nearby cells, mesophyll cells, and these chlorophyll molecule absorb it and give to the reaction center. And reaction center give the electrons to primary receptor. So in the Z scheme you can see this is the P680, the reaction center, PS second. When the red light comes in nearby cells, then it goes to the reaction center, and from the reaction center the electrons goes to the pheophytin. Pheophytin is the first electron accepted. Then from the pheophytin it goes to the plastocyanin, plastoquinone PQ, and from the plastoquinone it goes to the cytochrome B to F, and during this a ATP molecule is formed from the ADP molecule, and from cytochrome F the electron goes into the plastocyanin. Okay. Then from the plastocyanin the electron goes to the P700. P700 is the photosystem first, and from the P700 the electron goes to the FES, then ferredoxin and FNR, and during this one NADPH2 molecule is formed. So this is the Z scheme or non-cyclic photophosphorylation, and the second one is cyclic photophosphorylation. What is cyclic photophosphorylation? In this only PS1 takes place, only PS1, that is P700. So it consumes the sunlight and release electron. Electron is accepted by the ferredoxin. Through the ferredoxin the electron goes to the cytochrome B to F, and during this ATP molecule is formed, and from cytochrome F the electron comes to the plastocyanin. Then from plastocyanin it again goes to the P700, that is reaction center. So this is the cyclic pathway, and during this NADPH2 molecule is not formed, only PS1 is working, and Z scheme PS1 and PS2 both are working, and with the ATP molecule NADPH2 is also formed. So it is all about the Z scheme and cyclic photophosphorylation.